Jonathan Sebat. I'm a professor of psychiatry, cellular and molecular medicine at UC San Diego. And to my, that would be my right, although I'm actually pointing to the left, is Marieke Klein. Introduce yourself.
Hey, I'm a postdoctoral researcher working in Jonathan's lab. Started earlier this year, actually, on the CNV project. Did my PhD on ADHD genetics mainly, and now diving into the combination of rare and common genetic variants.
So we're co-presenting this presentation today. I'm actually just a freeloader, riding on Marieka's coattails, presenting some introductory slides, and then we will get into the meat of the presentation and switch presenters. We'll start with, forgot to mention. Those of you who, so we've left lots of time for the Q&A. So if you want to ask a question verbally, we'll be monitoring the participants. So click on participants directly. You'll see that you can raise your hand and click raise your hand, and we'll unmute you and you can actually ask your question directly. And alternatively, if you don't want to do that, you can type it directly into the Q&A box, and we'll be monitoring that as well. So we'll field questions afterward, and feel free to ask it any way you want. I'm going to go ahead and share my screen. All right. Able to see everything, Marieke?
Perfect.
Okay. So the PGC CNV group of the Psychiatric Genomics Consortium has been busily processing and analyzing data for a series of psychiatric disorders. And today we're going to present for you the overall analysis strategy and the preliminary results from the combined analysis of multiple psychiatric disorders. This is a collaboration of multiple groups within the PGC, as well as making use of data that is being analyzed in a separate consortium called the Genes to Mental Health Network. The goal of that consortium is to really understand what psychiatric traits are impacted by rare variants and polygenic scores, and to try to tease apart how the nature of the gene action in psychiatric disease. And so the PGC and the G2MH have a lot of common goals in mind. So this was a good opportunity to stimulate that kind of collaboration. So the team involved includes Marieke, Omar, and Juan from my team at UC San Diego. Let's not forget the rest of the PGC CNV group, including Steve Scherer's team, Jeff McDonald, Buma, Sarah Varandapram, and as well as Mount Sinai, Alex Charney, and Agat Dapin. Of course, this couldn't have been done without a lot of help and cooperation from the PGC disorder groups, both including ADHD, autism, bipolar, major depression, PTSD, and schizophrenia. In addition, other members of the CNV group include Sebastian Jacomont's group, and the latest addition to in terms of a disorder group has been groups that are collecting clinical array data through clinical genetics laboratories. And so we've had a great collaboration with commercial laboratories like LabCorp. So Stan Latovsky and his team have been huge contributors to the data sets and the analysis strategy that we're talking about today. So CNVs, as I think most people watching this presentation know, are some of the largest effect rare variant risk factors that we know of for psychiatric disorders. CNVs have been linked to multiple neuropsychiatric diseases, and they contribute in part to the shared genetic etiology of these different disorders. It was actually copy number variants were really the first evidence that autism and schizophrenia have a shared genetic basis, because that was there where we first got the glimpse that the same CNVs were impacting multiple developmental and psychiatric disorders. And that's now been proven to be true across the full spectrum of psychiatric risk, both polygenic and rare variants. And so these pleiotropic effects of the CNVs are ultimately going to be a key insight into how do genes relate to psychiatric traits, because the rare variants are one way of isolating a single allele and looking at how that allele impacts specific traits quantitatively, and how that, combined with other variation, impacts the traits in a complex fashion. So the rare variants are actually going to be a very useful tool in understanding complex genetic inheritance of psychiatric traits. So what we're talking about today is sort of what is the risk of the CNVs in terms of CNV main effects across a range of psychiatric disorders, and also how do CNVs interact with other genetic modifiers like polygenic risk. This was our flagship project, published in 2017 when the CNV group first got going, and of course the most intrepid of disease groups was the schizophrenia disease group. We're able to make some rapid progress early in the PGC's CNV group work that resulted in a CNV study of 40,000 samples, which was the schizophrenia CNV study. That was really kind of established the pipeline for the analysis that we're talking about today, where we were able to identify 8 different CNV loci that carry significant risk for schizophrenia, and also at the individual gene level, use that information to identify gene networks and pathways that are related to psychosis risk. And so now that first study was quite an experience. It kind of laid the groundwork, but we've learned that you can't necessarily do each disorder exactly that way, and you can't just do them one at a time, because this process of collecting all of the intensity data and then linking up the intensity data with all the imputed genotypes that are elsewhere inside of the PGC's databases, and then processing everything from beginning to end, that is a very time-consuming process. Both time-consuming in terms of getting the data and linking everything up, and then there's an entirely separate issue of data access that has to be handled. So the combined of all of that, you couldn't get all of the disorders done if you did them one at a time. So the CNV group really got every disorder started at the same time, so that we could go through that whole process and reach the finish line within the PGC's grant period. And we're happy to say that we've got all of these disorders shown here through the pipeline, with the exception of with the major depression being sort of the one that's just reaching that point now, but all of the others are completely through the pipeline. That pipeline consists of both collecting the data, processing, CNV calling, QC, and then association analysis. Now, the PGC is evolving, and so the structure of things so we're now reaching the PGC 4 grant. So the 4th phase of the PGC's NIH support is going to have a different model for actually putting the data through the pipeline, where stage 1 analysts within the PGC are no longer going to be just grabbing the genotypes and without the intensities, which was how things were done originally. So now all of the data is going to come through the stage 1 analyst in the beginning, and that will then be split into the CNV and GWAS pipelines. And so whatever young assistant professor who's looking to take over the CNV analysis, when I retire, which will be retire from this particular position, will hopefully this pipeline will be pretty much established and be much less labor-intensive process. I won't get into the nitty-gritty details of the individual calling pipelines here, but just to explain the details, that AFI and Illumina platforms have slightly different calling pipelines involved, and also AFI 6 and AFI 500 are a little bit different. So there's no single pipeline, right? There is an optimized pipeline, a variety of different ones that can be used depending on the platform. And so in the end, the name of the game in the end is not trying to process every array in an identical fashion. That's just not possible. The key is to take this diversity of different platforms and slightly modified pipelines for each platform and try to bring it all together in the end and do a meta-analysis in a way that will give you, where you can control for platform differences in your tests for association and get reliable results out the other end. So really, the philosophy here has been to allow for a certain level of chaos in the beginning, and then you try to tame that chaos at the end by taking a careful look at where your sources of platform-specific effects, case control imbalances in certain sections of the data. You want to find out where are your sources of bias and try to control for those. That's really the name of the game in these big heterogeneous data sets. So the data sets that we're looking at now, there's a combined total of about 400K samples that have been through the pipeline. And that includes ADHD, autism, bipolar, schizophrenia, PTSD, and clinical genetics laboratory data sets. And there's another batch of data that's also been through the pipeline also and are soon to be added to the data set, which includes sections of the UK Biobank that were not previously included, as well as major depression. So the combined data set that we're looking at will be something in the ballpark of about 700,000 samples. For the cross-disorder analyses that you're going to see today, a majority of the effort was focused on CNVs for which platform differences and other sources of bias are not a major factor. Large recurrent CNVs that are easy to genotype on all platforms. So the cross-disorder analysis in the early stages really tried to avoid the real difficult details and focus on the most clear risk factors. And so what Marieke will be presenting will be mostly focused on those. And also these are variants which generally don't have the same level of population stratification that you have with common variants. And so if a subset of the data set didn't have genotypes available at the time, it was okay to go ahead without, leave a subset without principal components and things like that. So there were a lot of advantages to focusing on the rare de novo recurrent CNVs in the early analysis. But the final analysis is going to have principal components, polygenic scores, and CNV calls across the full 700K, or at least a vast majority of the 700K samples. So the analysis plan moving forward, overall, we want to get an assessment of CNV associations, novel CNV risk factors, both individually within disorder, but also what novel CNVs can you discover if you combine all disorders into one and actually analyze the full 700,000 combined. And then, of course, we want to also understand in more detail what is the nature of these CNV effects. What is the risk profile? What disorders is a particular CNV associated with? And how does that interact with genetic modifiers, which Marieke will be presenting. Just some quick snippets before I hand the presentation over to her. Caroline Nievergelt and Adam Mayhofer have really done a fantastic job with PTSD. We were really excited to be able to include a disorder which is environmental in nature, and where maybe looking for the rare variants of large effect may not have been the highest priority of the CNV group, but Adam was not going to stand for that, and has actually really pushed PTSD forward. And he has CNV calls, QC on 167K samples, and has completed the overall burden analysis of PTSD. And so our, you wouldn't predict in advance that there would be a very striking rare variant effect in a disorder that's principally caused by environmental trigger. But Adam is happy to inform you that rare variant associations are in fact a factor in risk for post-traumatic stress disorder. And so this has a significant burden effect, particularly for duplications. So that's another interesting fact that that one might have predicted, that the deletions of largest effect may have less of a contributing role in PTSD, whereas the duplications of slightly more moderate effect might be the rare variant factors that might have a bigger role in this particular disorder. And it looks like the duplication burden effect is significant in PTSD. Thank you, Adam. The combined analysis, because of some minor details and getting access to genotypes and various different data sets, it wasn't possible to have all the genotypes and all the CNV calls together all in one pile in the beginning. And so the cross-disorder analysis that started early on was on a subset of samples where you could easily match up the calls and the principal components in the genotypes. So, and this was obviously, we're not intending to publish data, a subset of the data. This was done specifically with the intention of going through the whole full pipeline. And there's an important difference between a CNV association study and a GWAS, and that is we don't have imputation. We don't have a means of making sure all of the same variants are genotyped across in a consistent fashion across all samples to where it's easy to combine everything into one data set. So without something like imputation as an advantage, you're guaranteed to have variants that are present in one sample and not another, and case control and balance and factors like that can contribute to spurious associations. And so you're going to see spurious associations when you run your analysis. And so we really, it helps us to be able to identify the platform-specific effects. So platform-specific associations are something you want to see early on so you can identify them and then control for them. So we're happy to see in our cross-disorder analysis the largest effects come from some of our familiar large effect variants like 22q11.2 deletion syndrome, 16p11.2 duplication syndrome are still the largest effects. But as you can see here, there's a lot of other loci that come up, and some of you will be looking, what on earth? I don't remember there a 3p26.3 syndrome. I don't remember a 10p12.31. And there's a reason why you don't know about those, because they're probably an artifact. And so this preliminary analysis gives us both true associations, and it's also, we can easily spot artifacts that are attributable to platform-specific differences. So these kinds of efforts help us in working out the pipeline. And then when you move forward to the full set of 700,000 samples, you have these biases under control. So in terms of next steps, CNV association analysis to be done for individual disorders, novel CNV risk loci discovered from the combined sample, and then the cross-disorder analysis in more detail, understanding genotype-phenotype relationships. And so now I will go ahead and start to hand over this presentation to Marieke for her part. For those of you who want to ask a quick question while we switch gears, I can certainly handle that. So feel free to raise your hand or type into the Q&A. And I'm going to stop sharing my screen. You're all set, Marieke? There's one question in the Q&A which I'll address now. So David Curtis asks, how sure are we that CNVs contribute to ADHD specifically, rather than to some degree of intellectual disability? Do CNVs definitely contribute to people with ADHD and normal IQ? Do they contribute more in people with ADHD and lower IQ as for schizophrenia? Do they contribute more in people that have the combination of ADHD and intellectual disability, David says. Well, David, that's actually something we will touch on again, probably at the end of Marieke's talk, but I could address it briefly here, which is the CNV main effect obviously has a distribution, whereas a subset of CNV carriers will have intellectual disability, and a subset of them at the other tail of the distribution will have normal IQs. And so, there's a main effect on And then the question is, on what, right? So it's going to have a main effect on IQ. It's going to have a main effect on social responsiveness. It's going to have a main effect on, if you have dimensional measures of psychosis, you can measure the effect, you can measure all of the main effects, and then start to understand at the level of the individual CNVs, at that specific allele, how is it impacting psychiatric traits versus how is it impacting intellectual capacity. Also, are the relationships of copy number to these individuals' traits, so the correlation of copy number with these individual traits, do you have the same relationship with psychosis risk as you do for intellectual disability? And that's precisely the question, David, that I think Marieke is really tackling at the moment, is to understand where are the major effects, what traits are they impacting, and do you have the same relationship to psychosis risk as you do to IQ? And that's really the open question. And you know, it very well may be that some CNVs are really primarily impacting intellectual capacity, and the psychosis risk is a very modest effect. And there may be other CNVs that look differently than that. So that's our one Q&A. I'm just taking a quick glance over at the participant screen to see if we have a hand raised, and we so we'll go ahead and take it away, Marieke.
Okay, thank you, Jonathan. Yes, so indeed, I would like to present some preliminary results that we have so far on the analysis of genomic CNVs across the 5 psychiatric disorders that we have included at the moment. So the first part of this presentation addresses the first question that we had, basically, what is the risk of those pathogenic CNVs across the diagnostic categories, so across the 5 disorders. And to answer this, I'm going to study 93 recurrent pathogenic CNVs, and I will present results on 42 multigenic CNVs that have at least 50% overlap between the CNV call and the target region. In the future, we will also aim to include the single gene events, but we have to wait for another PGC Worldwide Lab meeting for that. So here I can show you like the samples that we included so far, where you can see that for ADHD, the sample size is still relatively limited, but for autism and bipolar, we are quite good. And this data also, these results also include data from the clinical genetics labs. So these are mainly participants with developmental delay that have been seen in a clinical diagnostic setting. So in total, these results were based on 260,000 samples. And this first plot here shows 8 examples for loci, in which we compared the frequency of cases versus the combined set of PGC controls. And you can see still for ADHD, what I just mentioned, the data set is still relatively small. With this green band, the confidence intervals are pretty wide. And for bipolar, as an overall pattern, it emerges that there is not that much going on in the orange bars here for most of the loci. But we also see that there are specific patterns of associations for different loci. For example, for the 3q29, we see that, for example, the deletion seems to be a risk factor for almost all disorders, whereas the duplication seems to be more specific for developmental delay, or basically what has been ascertained with the Klein sample, but not so much for any of the other disorders. And this is similar, for example, for the 22q11 deletion and the duplication pattern. So this, gives an idea of that not all pathogenic CNVs confer the same risk to all the disorders. There are differential effects for different loci, and there are also differences, differential effects between deletions and duplications. So basically that those risk variants, they can have certain risk profiles specific for the disorder. Something else that we did with these effect sizes for the CNVs is to see if we can determine whether there are specific CNV loci that cluster across the different disorders. And for those, we used hierarchical clustering analyses to see whether there are sets of loci that tend to cluster across disorders. So what you can see here is that kind of dendrogram, and I marked 6 main loci that emerge from these analyses. So for example, yeah, it's maybe a bit hard to see, but what you can observe is that not all deletions and duplications from the same locus, from the same CNV cluster in the same CNV cluster. So there are really some differential effects. And something that we want to investigate further is, for example, if the gene sets that are involved in individual clusters, if they differ, for example, in terms of their brain gene expression, and we will do that in collaboration with a PsycEncode working group. And so in sum, what you should mainly get from this dendrogram is that we see there are sets of CNV loci that tend to cluster together, but actually we are of course more interested in how this behaves across the different disorders. So that's how we address this question in the next slide, where you can basically see on the left of this plot the dendrogram again, and then we plotted the effect sizes for the different disorders in a heat map manner, basically. So what you first observe is that the disorders, they cluster according to the period of their onset. So for example, autism and developmental delay or intellectual disability, they seem to cluster closer together, whereas the later onset disorders, bipolar, schizophrenia, but also ADHD, they seem to cluster more closely. What I think is striking is that ADHD even is closer to schizophrenia than bipolar is, but again, we need to keep in mind that the ADHD data set was one of the smallest, so the effects are overall a bit lower, and this is also reflected in the colors of the heat map, where red indicates the highest effect size and blue the lowest. What we also observe is that the red part here from developmental delay is kind of the most dominant cluster that we observe. To a lesser extent, this is also true for autism. But if we look into this a bit more specifically, then we can see that there's one very distinct cluster, and this is actually significantly the most distinct cluster, where the CNV effects are bigger in bipolar, schizophrenia, and ADHD compared to autism and developmental delay. So this kind of method can help us to group CNVs based on their phenotype associations. And in the future, we will include these single gene effects as well. We also want to include the PTSD and MDD data. And I think what is also very interesting, and what is a bit related to the question that we just discussed, if we include data from the UK Biobank on, for example, dimensional traits, something like cognitive functioning, how these clusters will then be consistent or change, and how those patterns will look like. And again, we will also further investigate characterize the clusters and compare them those in terms of, for example, PLI scores, see if there are brain expression differences, for example, for this specific cluster compared to the others. So this is still something that we have on our schedule to do. What I would also like to show, and what we used for this effect size data for, is that we can see a differential relationship between the copy number and the traits, specifically for intellectual disability and schizophrenia. So what you can see on the left is the effect sizes between the deletions here on the x-axis and the duplications on the y-axis. And you see for developmental delay that there's a strong positive correlation, whereas for schizophrenia, the effect sizes for those deletions and duplications are overall negatively correlated. And this is a general pattern. This is not specific for 22q11, which you can see over here. So even if we take this one out, we still have, so this correlation is attenuated, but it's still persistent. So what this basically leads us to is these differential patterns of gene dosage and trait relationships. They can, so they are very different depending on the different traits in question. So this is one example to simplify this a bit for the 22q11 locus, where you basically see for intellectual disability or developmental delay, you see a U-shaped trait where the deletion increases risk, but also the duplication increases risk. However, for schizophrenia, this is a different pattern where the deletion increases risk, but the duplication actually is protective. So we now are able with the combined data to observe such trait gene dosage relationships across different disorders and also across all different loci. So I think that is very intriguing, and we are definitely following up on this, and yeah, by enlarging our sample sizes basically to have enough power for the individual loci. So something else that we are interested is in this, what Jonathan mentioned briefly, is the variable phenotypic expressivity. So what this table basically shows is the average frequency of neurodevelopmental phenotypes for selected variably expressive and syndromic CNVs. So you can see that not each CNV actually increases the risk for, confers risk for 100% for each disorder. So there's a lot of variation in there. And also, even when a phenotype is present, these can range from pretty mild symptoms to more severe syndromic forms. So the second question that we want to address in this project is basically which factors determine this variable expressivity. And one possible mechanism to this variable expressivity can be the contribution of common genetic variants as measured by polygenic risk scores. And these effects of polygenic risk scores in CNV carriers, they have been previously reported, and I'm showing a few examples of those here. And what main conclusion from these publications is that the PRS effects, they can actually contribute to both the increased risk for a certain disorder, but they can also contribute to this variable expressivity of dimensional traits, either within a disorder, but also across disorders. So with our combined data set, we are now able to analyze this more consistently across multiple disorders and across many different loci. And in collaboration with other groups, we are able to increase the sample sizes for specific CNV loci to analyze those effects. And one of those collaborations that I would like to mention between the PGC and the Gene2MH, the Gene2Mental Health Network, is addressing one of these key questions in which we aim to analyze this additive or maybe even interactive effects of rare and common variation on behavioral dimension in CNV carriers. So this network set out to collect dimensional measures on neuropsychiatric syndromes, and I think they started off with doing that for 2 loci, the deletions and duplications, but actually we are getting more and more on our list. And then to examine the convergence and the specificity of these phenotypes across the different CNV carriers. And we hope that this will increase understanding of the variable expression of these dimensional phenotypes in carriers with the same pathogenic CNV. And we will also analyze the additional genetic factors, both rare and common variants, and some environmental factors as well. And especially project 2 of the G2MH network is involved in this collaboration, which includes several sites from the US, Canada, UK, and Europe. And I listed a few of the most people who are our main collaborators on that. So one example of such a combined effect of a copy of a CNV and a PRS on a dimensional trait is something I would like to show on the example of the 16p11 locus. So for this, we used the Simon's VIP dataset, which included around 260 individuals. And we used BMI as a dimensional trait in this example. And what you can see is that on average, the 16p112 deletion carriers, they have a higher BMI. But also what we observe is that in controls and in duplication carriers, a high risk or high PRS for BMI is associated with an increased BMI Z-score. And this suggests a modifying role of common genetic variation in addition to the CNV effect. However, for deletion carriers, we do see a similar trend, but this is not a significant effect. We also have to keep in mind that this is a relatively small data set, and this is only nominally significant over here, but it's maybe you can just see it more as an illustration of how we would like to approach this. And again, with the larger data set from the PGC and our collaboration with G2MH, we are able to do this more consistently across multiple disorders and the different loci. Yeah, hope to contribute to that in a more systematic manner, actually. So this brings me to the conclusions already. So we saw that these CNVs tend to cluster based on their phenotype association, which suggests that specific CNV alleles have distinct psychiatric risk profiles. We saw that some clusters are grouped by being predominantly developmentally lay and autism, but we also observed a significant cluster which seems to have bigger effects, especially in bipolar and schizophrenia. And in the future, we will test for enrichment of biological processes and gene expression of individual clusters to better understand the biological mechanisms within these specific CNV groups. We will also expand the range of the cognitive and neuropsychiatric disorders and traits by including additional CNV codes from other PGC disorder groups, but also related phenotypes from the UK Biobank, most likely in the cognitive domain. And we will also investigate the modifying role of common genetic variation across CNVs and across disorders. And this basically brings me back to the acknowledgement slide that you already saw. So yeah, special thanks to the group in San Diego and the PGC working groups, the lab from Sebastian Jacquemont and our collaborators at LabCorp. So with this, I think I will stop share my screen, and I hope that we have more questions in the Q&A. And I think I will hand that over to Jonathan again to moderate a bit through the discussion.
Sure. So I'm able to see a couple faces on the screen here. If there's anybody who wants to be unmuted, just click on the participants button at the bottom of the screen. Don't click on the arrow, click on the actual icon, and it'll open up a participants bar. At the bottom of that bar, you'll see an option to raise your hand. So if you click on that, then we can unmute you if you want to be. So I will go ahead and we got some new stuff in the Q&A, and then we also have some, I was having a good time in the chat box during Marieka's presentation, so we can touch on that too. So the second question is, can you talk, Marieke, I'll let you start taking the questions. Can you talk about progress in moving from CNV effects to specific gene effects and biological mechanisms?
Is that a question in the chat, or is that some? I'm sorry.
That's in the Q&A. If you click on Q&A.
Oh, there I see it. Okay, thank you. Yeah, I think it's. I think we can. I mean, the results that I presented, of course, they are preliminary, but there will, with the biological follow-up that we are planning to do, I think we will be able to do so. So to see if there are certain genes within each CNV that are more risk conferring than others. I think that should be definitely possible. Yeah.
There's another collaboration that we're trying to kickstart, which could help on this question, which is PsychEncode. So there's probably plenty of PGC-ers that are involved in PsychEncode already, and we've had the pleasure of chatting with Dan Geschwind and Mike Gandel. And this is, there's a lot of new data coming out on single cell, RNA-seq, postmortem brain, in schizophrenia, bipolar. And so I think we can start to look at the question of, so if you have a specific set of trait relationships that you've nailed down to a single CNV, that makes it unique. Or maybe there's a cluster of 5 CNVs that have a similar phenotype profile. What do they have in common, both at the pathway level, but also at the spatiotemporal gene expression level? Okay, so is there a cell type or a developmental pattern of gene expression that might correlate with your phenotype profile? So that would be one way of getting at this question. So I think the interactions between consortia is actually so So psychiatric genetics is no longer just a big consortium effort. It's now a consortium of consortia. But the interactions between these different consortia that are working with different data types, I think that's the key to getting at these biological mechanisms. So Marieke, your second question comes from Stan Litovsky, and he asks, any concern on confounding between platform and disorder?
To be honest, I haven't checked it for each of the loci and all of the calls. What we overall see is that at least for the a bit more frequent variants that we analyzed, there is not a single platform that we are missing or that is dominating the calls. So it's a good point to check that more general, but I think also for these recurrent CNVs, they are very robustly assessed across all the different all the different platforms. So I don't see that this would be something that is severely confounding the results at the moment.
Yeah, Marieke, your results are focused. I mean, you specifically selected the large recurrent CNVs, which don't have much population stratification and can be detected by nearly all platforms. So that's but that doesn't necessarily mean that there isn't any effect. There could be differential sensitivity that we maybe haven't fully evaluated. So there may be. I think we could start to look at the question of whether there are platform effects, even with a megabase size deletion. We maybe have the ability to detect platform effects there and hopefully correct for them. And then in my part of the presentation, I actually specifically highlighted examples that I suspect are platform specific artifacts. So we fully expect there's no way to completely eliminate them in advance. You actually have to discover them empirically. So you actually run a specific analysis to test whether deletion counts or duplication counts are very skewed at a specific locus. So go probe by probe across the genome and ask, are deletion counts or duplication counts skewed very heavily to one platform? And actually find these platform biases and account for them. So that's the strategy that we hope to take. And Stan happens to be very familiar with the Cytoscan HD platform that all the clinical labs are using, and that has the biggest issue because it is a 4 million probe array, which is much more dense than any other array. That it's also the most non-uniform in terms of its probe coverage. So I think that the Cytoscan HD clinical arrays are going to have the biggest issue, and that's one where we may need to actually handle that data set with very great care, and perhaps leave it as a secondary data set rather than a combined sample. It depends on the analysis.
Yeah, one thing also to mention is maybe that for for the different disorders, they were all run on different platforms, right? So there is quite a distribution of there. So it's not that we run ADHD all on FE 500 and schizophrenia all on Illumina. So we didn't break it down in this presentation, but it's quite diverse within each disorder. So I think that also takes away a bit of the concerns about confounding.
So we still haven't gotten to the end of our hour, so I can indulge ourselves a little bit more by diving into the chat box. How do I open that thing up? Click, there it is. So I think it began with Danny Weinberger asking a question about in the early my part of the presentation, we talked about how the IQ of a CNV carrier can vary between being intellectual disability and being similar to the average IQ in the population, having an IQ of 100. And Danny's question was, isn't the concept of IQ being normal, or like the fact that there's an average IQ in an individual, isn't that concept somewhat misleading? If a CNV carrier has an IQ of 100, but the SIBs and parents of that individual all have IQs of 120, is that individual normal? Right? There's may very well be a CNV effect, and I completely agree with Danny's point. It's absolutely the case that's the nature of the psychiatric trait, that you have a combination of rare variant effect and common variant effect that's acting, and it's acting in every person. It's not as if the CNV has a muted effect in one person and it has a strong effect in another person. The effect may be very similar across individuals, but the background polygenic load may be differing between those individuals. Maybe I'll pull up the BMI example really quick, because I think that is really instructive in this case. I'll go ahead and share my screen again and use the presentation. So that's not BMI. Where is it? It's the BMI example. So I think this is a good one, because this case, there is a CNV main effect. We don't necessarily assume the effect of the CNV is different in all of these different individuals. The reason why the BMI is spread so broadly, the deletion carriers and the duplication carriers, is because of the polygenic load. So you have this, in fact, the polygenic risk has a very similar effect across all CNV genotypes. So you have a very similar effect. So it looks like a classic example of an additive effect of common and rare variation. You have a very similar polygenic effect across all of the rare variant genotypes, but the rare variant, so the deletion increases your body mass index by about 0.7 standard deviation. The duplication decreases the average BMI by about 0.5 standard deviations. So I think maybe when we start to look at IQ, maybe IQ will look similar to this. Here's the one difference is that IQ doesn't move in a linear direction in this case. IQ goes down with both del and dup. So you're going to have a slightly different effect if you look at IQ. So both the deletion and duplication will be reduced. And then there will be polygenic scores for educational attainment and other factors that explain variability in IQ. And so, but it's the same general principle that you're looking at here. There is a CNV effect, and it's probably having an effect in everybody. We have a new addition to the Q&A, so I'm going to zoom in. Stan Latovsky asking, the plots showing schizophrenia having a monotonic effect on gene dosage versus developmental delay having an optimum value is puzzling to me. I can see different genes having a monotonic versus bowl-shaped response, but why all genes for a given disorder? Shouldn't they vary gene by gene within a disorder? Stan's raising his hand. Thank you, Stan, because that is
I figured out how to unmute. You read the question.
Okay, why don't you ask the question again? Because that's a mouthful.
Can you, Marieke, can you go back to the slide with the dosage effects?
Yeah, let me just get that one.
Yeah, and I think if you have an animation or something.
You had that third animation,
yeah. All right. So the picture, the inset, is two different ways a gene can respond to dose, right? It can respond monotonically, like the purple, right, the trait, or it can have an optimum. So in this case, the green is tuned to a particular value. If you depart from that value either direction, it's bad. And the other one is a sort of standard dosage effect where, you know, if you have less dosage, it's bad. So we understand that on it, that different genes may respond differently. Some of them may have an optimum value. Some of them don't care as long as there's enough. But why would all the genes for a given disorder fit exactly one of those two categories?
That's actually a really good. Oh, sorry, Marieke. I think he's asking you.
Yeah, I was going to say that I'm actually not sure if it's really all genes, right? So this is an overall pattern for all the different loci that we see here. But there will be exceptions from the rule, right? So for example, for 16p11, we know that this is not actually happening. So yeah, I'm not sure.
Maybe another way to ask the question is there a way to ask this question on a gene by gene basis rather than for the whole disorder?
Yeah, I think I could see possibilities to try that.
Yeah. Like Evan Eichler, you know, years ago had this genome-wide dosage map, you know, where they looked for all genes that had a dose-dependent effect. Maybe there's a way to do something similar here, where you identify which genes, you know, have a monotonic effect versus a bowl-shaped, whatever you call that, a green effect.
Yeah, I think that is an interesting idea. I think especially there's a few CNVs that have like really a lot of genes included, right? Whether others are relatively small and only have one or two. So looking at that in a gene-specific manner, that may be quite elusive. Yeah, that's a good suggestion.
I also think we might be. You know, we might be generalizing. We might be overgeneralizing here, right? If you look at the negative correlation for schizophrenia. So let's actually dissect these a little bit. On the left, you have intellectual disability, and the reason, presumably the reason why you have a positive correlation of deletion effect size and duplication effect size is because it's the nature of the genes. You know, it's the nature how dosage sensitive are the genes, and roughly how many genes are there, right? That's ultimately determining the effect size of a CNV for intellectual disability. And it doesn't matter if you have a deletion or a duplication. It's the nature of the underlying genes that's determines how strong of an effect you have on you know, intellectual disability. So that's that trait. And schizophrenia is going in another direction overall in general, but it may not be as tight of a correlation, Stan, right? There may be. It may actually be really 22q, 3q29, 15q13. Maybe those, you know, the ones at the extreme lower right and upper left are the ones where you have these reciprocal effects. So presumably there must be an overrepresentation of genes that are impacting a particular biological process. It may be a similar biological process, and so it's affecting the copy number is affecting that biological process in a directional manner, and that's how you get these contrasting effects between del and dupe. And in some cases it's the deletion that's the risk allele, and in other cases it's the duplication that's the risk allele. That doesn't matter. The main point is that there's a very big difference between del and dupe for schizophrenia, but that's not true for all loci, right? If you look in the middle, there are plenty of loci where it's certainly there are some loci where both dupes and dels are risk. So like 17q112, 2q11.2. Those, you know, it may be a totally different situation there. If you could actually measure the quantitative effect of copy number on traits, they may not have the same relationship that the other loci have.
I wonder if it could be a thing about developmental stage, that maybe the genes active early in development are more finely tuned. And
that's a great hypothesis. Yeah. That's a hypothesis that can be tested. If you could predict, you would hypothesize that really early fetal gene expression. So if you could rank the CNVs based on how early in development the dosage sensitive genes are expressed, maybe the ones that are really, really early in development are having a big effect on intellectual disability and maybe are not having a linear effect on psychosis risk as much. Whereas something that's expressed in a different cell type or later in development, maybe there's a particular process there that's a little bit more specific where you could have that directional effect on psychosis risk. That's a naive hypothesis that I'm not saying your hypothesis is naive. We've thought of the similar thing. It's something that could be tested in for example, in the psych encode data. It looks like Ed Cook has a question. So I think, Ed, are you unmuted?
Yeah, just a quick follow-up. Interesting discussion there. I was just thinking about with CNVs, the relevant gene could be different in a deletion and a duplication possibly, or more than one gene in a large CNV.
Yeah, I think that's definitely true, right? So that's why we see these kind of differential effects between deletions and duplications. And I think that the idea from Stan to look more into the gene-specific relationships, that can really help us in disentangling this. Yeah.
So I wonder if you, if under that kind of a model, Ed, if it's actually one or two genes over here that are really relevant in the deletion, and then one or two genes over here that are relevant in the duplication, then the question, then would you expect to see the inverse correlation with body mass index, for example? Would you expect a deletion to increase BMI and a duplication to decrease BMI if it's different genes? Would we predict that, or would you? So that there may be particular patterns that might be indicative that there's a common gene set acting in a quantitative fashion, and there may be other patterns that would be consistent with your second model, right?
Yeah.
The other patterns would be, yeah. The other patterns would be that you would have, you wouldn't necessarily see the effect size wouldn't flip. Maybe the effect size won't flip between DELs and dupes, but you would really just have a completely different risk profile for the DEL. The DEL and the dupe would have an uncorrelated risk profile. Whereas if it was a common set of genes, maybe you would have an anti-correlated risk profile. So you would have a strongly anti-correlated risk profile versus an uncorrelated risk profile.
Would you predict that a larger CNV would be more likely to be bowl-shaped because it's got so many genes, one of them's got to go wrong, right? So you move in either direction, you're going to mess up somebody. Whereas for a small CNV, maybe only one or two genes, only one of them phenotypically important, most genes probably monotonic dosage effect, then you would be on the non-bowl-shaped trajectory.
I think, I mean, I'm sure that some loci do, both DEL and dupe do increase schizophrenia risk. Like 1Q21 is an example, right? Where both the DEL and the dupe increase schizophrenia risk, maybe not to the same extent. So there you have it. There is a U-shaped relationship there, and some CNVs have it. I just don't know that it's going to necessarily correlate simply with the size of the deletion or number of genes or something like that, because 22q is actually one of the largest deletions in the set, and that's the one that has the strongest protective effect for psychosis. So it may not be the size necessarily, but it will probably be the genes. So some genes will have a U-shaped relationship to psychosis risk, whereas others will have the 22q style monotonic relationship. And what separates them? That's a really interesting question. So like if you could imagine, if you could reliably really nail down the relationship. So some CNVs will have that monotonic relationship in a negative direction. Others would be in a positive direction. And in some cases, the DEL is the risk allele for some, and the dupe is the risk allele for others. If you could break these apart into their different relationships of dosage to risk, can you tease apart developmental expression patterns across those different functional groups, or say phenotypic groups, or different pathways, genes? I think that's the ultimate question. So as a consortium, what the PGC can do, and the Gene to Mental Health Network also, is really get the phenotype relationships nailed down, and then let the computational biologists go to town on data mining to understand what distinguishes this particular gene group versus the other.
Great discussion. I have to leave.
Thanks for joining us, Stan.
Yeah, bye.
We have more Q&A. So Marieke, I think David is back with a question of how much variability is there between CNVs at a particular locus? So between different, so I think you're talking about allelic heterogeneity at the same locus, like 22q. Are there loci where the boundaries tend to be pretty similar? Others which are much more variable? Always seems like a headache that they were somewhat different in size.
Yeah, so what I know is that there is variability, but I haven't analyzed that in with these regards to the current results so far. I think it's a good suggestion to still look into that a bit more, if there are disorder specific differences. Yeah, no, I only know, yeah, I know that there is variability, but it's for a few loci it's very consistent. For others, they are they breakpoints differ a bit. Yeah.
The breakpoints are inside of these very dense segmental duplication clusters. I think we're kind of SOL in terms of being able to really tease apart the allelic heterogeneity, because they're the arrays are just not up to the task of really getting the breakpoints down really clearly. At least not the arrays with the resolution that we're looking at. Whole genome sequencing does a little bit better, and then of course long read sequencing does far better. So I think one has to go into the Simon's VIP sample and the Genes to Mental Health Network samples and actually look in those data sets at the precise breakpoints and see, does the copy number of the segmental duplications have an effect in addition to the deletion and the polygenic risk? Is there an effect of the breakpoint? I think that's going to be interesting. And then of course there's the examples like Nerexin-1 where the breakpoints are completely random, and there, that's a completely different situation. It's a little bit more like an exome, where you have a whole wide variety of different alleles impacting the gene, some of which may be affecting risk and others less so. So that's a separate question. It's really at the level of the individual gene that you have to look at. So I think we might have gotten to all of our Q&As. So I really like to thank everybody for your for coming to the presentation, and I especially thank everyone for this really fascinating discussion. This has been helpful for us too. So that's really in the spirit of a lab meeting. So I'm glad that our worldwide PGCers could come and chomp on the bit with us today. Thanks so much, everybody. And I think that concludes our presentation for today. Thank you, Marieke.
Thank you, Jonathan.
And enjoy your weekend, everybody. Cheers.
Thank you. Bye.